I'm gonna introduce you, and then you're gonna introduce her, okay? Rolling introductions. Okay, let's get started, please. First of all, I'd like to welcome everybody. This is the kickoff for this year's Timothy Johnson seminar series. For those of you who don't know, Dr. Johnson, who's was a physiologist, was here at the earliest days of the start of the medical school and was one of the instrumental and key people in developing the curriculum here. And when I got to know Dr. Johnson, we talked a lot about the research domain and seminars we were doing and so forth. And he was very excited about this program we were going to start. So he tragically became ill and died soon after the medical school opened. So we named this series in his honor as the Timothy Johnson Medical Scholar Series. So this is the first lecture of this year. And we're having several others. As you know, you've heard a lot about the program from Dr. LeConte. And just by way of reminder, coming up in 2 weeks, we have another one. That'll be Dr. Hanchard coming from the National Human Genome Research Institute. We'll be talking about African human genomics. So it should be also a very exciting lecture. So it's my pleasure today to introduce the introducer for today's talk, and that's going to be Dr. Rob Gordy, who is the director at the Fralin Biomedical Research Institute of our Center for Vascular and Heart Research. And he, in turn, will introduce today's speaker, Dr. Grumbach. So I'll turn it over to Rob.
Thanks, Mike. Hi, everyone. Yeah, so there's a domino effect here. I'm actually standing with Steve Paulson, who unfortunately had another engagement that made him move away today. But so it's my pleasure to welcome Dr. Isabella Grumbach. Isabella is Isabella is Vice Chair for Research and Internal Medicine at the University of Iowa, and she's also a researcher in the Division of Cardiovascular Sciences and Internal Medicine at the University of Iowa, and also a professor of radiation oncology, which is a kind of an interesting combination. And we had a nice and interesting conversation about a radiation injury when we met early today. She trained in Germany at Ruhr University Medical School and also did a PhD there, and then went on to become a postdoctoral fellow at the University of Chicago and did a residency in internal medicine at Cook County Hospital in Chicago. And something that's not on the cheat sheet that Steve gave me was that in 1991, she was also doing a residency at the Medical University of South Carolina in Charleston. She'd come over from Germany for 8 months. And it just so happens, I was a British Heart Foundation fellow at the Medical University of South Carolina, visiting from London just about at the same time. So I said, we probably met, but didn't even know it. Who knows? But Charleston at the time was recovering from Hurricane Hugo, so a little bit of turmoil down there. So I'm not going to say too much more. I did discover this interest in radiation injury that we're also becoming more interested in, and I'm just really looking forward to her talk today. She has provided new insights into the mechanisms of vascular repair and remodeling in response to mechanical disruption, inflammation, viral infection, and has a particular interest in CAMK2 and mitochondrial
going a research program that I've lot of different ligations of metals and coated with an anti-proliferative compound will be left behind, and hopefully the blood flow is restored. And this actually works really well, except that in about 5 to about up to 20% of patients, there is buildup of another stent in the same area, and this is called in-stent stenosis. Diabetes is a very important risk factor for this, so this is about twice as common in diabetics, and about 10% of all angioplasties in the country are performed to treat stenosis. And for those of you who have learned about health economics, so the Medicaid reimbursement for this procedure is somewhere in the range of $15,000 to $20,000. When you multiply this by 500,000, then you know how big the burden to the healthcare system is. Okay, so balloon angioplasty in neo-intima, I just would like to introduce the term neo-intima because I was using this a lot. It really means nothing else but intimal thickening, as it's seen in atherosclerosis, but also in response to mechanical injury by balloon angioplasty. And this is kind of what it looks like. So this is a coronary blood vessel cross-section, and this is the coronary artery with the stent. You can see the struts here, and this is the neo-intima that has formed after the stent was placed. Now, importantly, one of the big players in both the buildup of the atherosclerotic plaque as well as the neo-intima is the smooth muscle cell. So in early stages of atherosclerosis, there will be some activation of smooth muscle cells, which sit here in the media. They are differentiated, they contract, and in response to exposure to growth factors or immune factors, the smooth muscle cells will start proliferating. And they do form a big part of the atherosclerotic plaque. And part of the reason why we are not studying atherosclerosis per se is not that we're not interested in it, it's that the animal models for atherosclerosis don't really focus on the smooth muscle cell aspect. They mostly focus on the aspect of the lipid accumulation. So really we study smooth muscle cell biology mostly in neo-intima formation, in part because the animal models are just so much better suited for this particular type of pursuit. So in neo-intima formation, there is a stent placement, and the endothelium will be injured, and the smooth muscle cells in the underlying medial layer will be exposed to growth factors again, and they start proliferating, and they lead to this buildup of smooth muscle cells, inflammatory cells, to narrow the blood flow, the lumen of the blood vessel, and limit the blood flow and cause symptoms. So the time course of this is actually very well understood, mostly from human, but also animal studies. So after the actual injury, after a few days, smooth muscle cell proliferation will strongly increase with a peak after a few weeks, and then the increased proliferation will subside. And on the other hand, there's some amount of apoptosis of smooth muscle cells leading to this buildup of neo-intima. And so importantly, from this slide, you can glean what the typical strategies are to reduce neo-intima formation, which are to, number one, most importantly, decrease cell proliferation, and on the other hand, to increase cell apoptosis. Importantly, for those of you who study muscle cells, smooth muscle cells are a little different from their big brothers, the cardiac myocytes and the skeletal muscle cells, in the sense that they are not terminally differentiated, meaning they can de-differentiate from this contractile state to a more amorphic fibroblast-like cell. And that is typically seen after in atherosclerosis or after injury. And these proliferative smooth muscle cells are somewhat different in their protein composition and mRNA, DNA expression. But these are really the types of cells which we're mostly interested in studying. And you actually would do this by taking a blood vessel, spreading the smooth muscle cells and growing them on a dish. So mitochondria and smooth muscle cells do interact or intersect in many different ways. First of all, proliferating smooth muscle cells have a reasonable amount of mitochondria, and the mitochondria serve a number of important functions in every cell, like in smooth muscle cells. So And in the aspects that we are most interested in are metabolism, loss production, and their effect on proliferation as well as apoptosis. So one of the key factors of mitochondrial function is uptake of calcium. So calcium is released from endoplasmic reticulum and taken up by this MCU, mitochondrial calcium uniporter, into the innermost area in the mitochondria called the matrix, where the calcium influx will increase TCA cycle activity. This will lead to more oxidative stress and lead to the production of reactive oxygen species or ATP. And on the other hand, the increased TCA cycle activity will also lead to more ATP production. And then mitochondria also play an important role in apoptosis. There is an association of back, which lead to the injury of mitochondria and their apoptosis. So the ways in which mitochondrial function intersects with new intima formation has been studied by a number of other people. Although I would like to say that we are the only lab studying this, of course we're not. So the other ways how mitochondria and new intima formation has been studied is by looking into leveraging loss production to regulate smooth muscle cell proliferation and migration. And in the last few years, a number of studies have looked into leveraging or manipulating mitochondrial metabolism to decrease cell proliferation. So the particular way how my lab is looking at mitochondria is by concentrating on the mitochondrial calcium uniporter or MCU. As I mentioned before, the calcium is released by growth factors via the IP3 receptor from the endoplasmic reticulum, and the mitochondria tend to localize to the vicinity of the endoplasmic reticulum, and the calcium is taken up through a pore in the outer mitochondrial membrane called VDAC, and then through the mitochondrial calcium uniporter into the mitochondrial matrix. And again, this then leads to the upregulation of a number of processes which are important for cell proliferation. So the mitochondrial calcium uniporter has been somewhat of an enigma. It's been known to exist for 50 years or so, but it's only really been identified on the structural level in 2011. And something of this importance then, of course, tends to be published in Nature. So we have two back-to-back papers from Vamsi Mufa and Rosario Rizzuto's group identifying the molecular identity of this gel. And as you can see here, so these are the number of publications in the last few years, and 2011 would be here, so the number of publications has gone through the roof and continues to be very high because of just the basic importance of this channel. So a few years, 2 years after the channel was identified, a group at the NIH actually came out with a mouse, right? Typical approach, you would knock out the gene in a mouse model, and to everybody's surprise, the mice were just fine. They're probably a little small, but otherwise, and there really wasn't any calcium going into the mitochondria. There's still some levels of it, but otherwise, the mice were running around just happily, right? And they didn't die. And even stressing them didn't really seem to have a big effect of this. And a lot of has been written about this, why this happened, and so forth. And some of this has to do with this particular mouse model that was made. And we can't really go into the details, but it's just a great example of something of the fundamental biological importance, which put in a mouse model doesn't seem to pan out quite as expected. Now, one of the reasons why, initially some of the studies about MCU didn't really show anything important was that it wasn't really known that MCU, actually that this channel is a complex. And so it has MCU, the main compound, right, which is the pore-forming channel. But there are a number of regulators which are very important for its function. They include a negative regulator, MCUB, and a number of other regulatory subunits. Importantly, those which are named like the medical intensive care unit, right, MICU1, MICU2. And so when the calcium levels at the outside of the mitochondrial matrix are relatively low, these regulators will actually block the access to the channel. When the calcium concentration at the outside are very high, they will kind of swing around and the pore will open and a lot more calcium is going to flow into the cell. So if you think about this, you do an experiment where you have mitochondria in a dish and you add increasing concentrations of calcium. In the beginning with low concentrations, there will be very little uptake because the pore is blocked. And then as the concentrations rise, the regulators will swing to the side, open access to the channel, and there will be a lot more influx of calcium into the mitochondria. So there's this sigmoidal-shaped uptake curve for this. Now, for example, if you then delete these regulatory subunits, the MICUs, what you will see is at low concentrations, there will be more uptake because the access isn't blocked any longer. And at high concentrations, there will be less uptake because the channel isn't as accessible as you would think. And if you take away the pore-forming subunit, well, there isn't very much calcium uptake. Okay, so it also has actually turned out that the composition of this complex is very different, whether you look at a smooth muscle cell, cardiac myocyte, or neuron. So there's a lot more to be learned about this. So in the next few minutes, we will talk about 3 different chapters. There'll be 3 small stories from my lab. 2 of them are published, 1 is about to be published. And we are going to talk about mitochondrial calcium entry as a driver of metabolic activity required for cell cycle progression. So really a story of how mitochondrial calcium entry drives cell proliferation. A second story about cell migration. So this is the positioning of mitochondria close to focal adhesions. It's required for smooth muscle cell migration. And then a last short story about why people with diabetes have increased cell proliferation, leading to a greater risk of new intima formation. And as always, this is the story of people in my lab, people who I collaborated with. So the first story really is the brainchild of Olga, who is an electrophysiologist who came about a decade ago from Ukraine and loves to work with cells. And so Olga started by just doing very simple experiments where she asked whether blocking or deleting MCU would have any effect on cell proliferation. And these were at the time when most studies about MCU came out were just negative, nothing was found. Okay, so here she did very simple experiments where she has smooth muscle cells and adds a growth factor called PDGF, platelet-derived growth factor, which is really clinically relevant for neo-intima stenosis. And she actually just counted the cells at different time points. And she found that either knocking down MCU, blocking it with an inhibitor, or using cells from knockout mice decreased the cell proliferation, but only when she added a growth factor. Right, so this is growth factor alone in control cells, and in red is MCU manipulation with growth factor. Meaning that in the presence of a growth factor, MCU inhibition blocks cell proliferation. And we also checked that the cells don't just die. That's the reason why there are fewer cells. So we did some apoptosis assays. But this really led us to really know with some certainty that the defect that we see really has something to do with cell proliferation. So then we embarked on a journey of sorting cells. And so here we, on the upper row, are wild type cells at different stages after starvation. And what we're actually staining for is DNA content. So in a growth arrested cell in G0 phase, there will be, all the cells will have about the same DNA content. As the cells start proliferating, more and more cells will make some variable amounts of DNA hoops as they progress through the cell cycle before they actually have doubled their DNA content. So that's why the second peak gets bigger and bigger as the cell cycle progresses, and then there are the cells in the middle, which are the ones which are actively making DNA. And so the actively making DNA phase is called S phase or synthesis phase. Okay, so as you can see, this wild type cells do go through the cell cycle. There are more and more cells in G2. And if you looked at 36 hours, the peaks would be kind of opposite in size. So this one would be bigger than that one, and the cells divide and go back to this phase. Okay, so in MCU knockout cells, however, the cells just start out very similarly, but then there are fewer cells entering into S phase, and the G2 peak never gets quite as big. So this led us to conclude that there is a defect in cell cycle progression starting in S phase, and there's a quantification of this. Okay, so we kind of were scratching our heads and wondering how to actually align this with MCU or mitochondrial function in general. So it was kind of our early days. And we were reading some really nice papers from a group at the NIH, which actually looked into mitochondrial shape during different phases of the cell cycle. So the mitochondria can fuse or they can fragment, depending on what the metabolic needs inside the cell are. So in a growth-arrested cells, most mitochondria will be little and fragmented. As the cell goes through the cell cycle in G1 to S phase, a lot more cells will be tubular or long, like this. And in G1 to S phase, actually, the mitochondrial ATP production in the cell is the highest, and this is to fuel DNA synthesis. Okay, so and then the cell will be ready to undergo mitosis, and for this, the mitochondria get fragmented, and the daughter, the fragments, then get distributed to the whole cell. So there are some cyclic changes in mitochondrial morphology throughout the cell cycle. And what Olga found was that in MCU knockout cells, this just never happens. The mitochondria are just little, and they stay little, and they never fuse. And well, what does this mean? Well, so this means that the cells actually also produce less ATP. So we found that, we and others found that depending on the cell cycle, there are differences in oxygen consumption rate by mitochondria to make ATP. In wild-type cells, the oxygen consumption rate goes up during the cell cycle, especially in S phase, to make more ATP. In MCU knockout cells, this just never happens. And this correlates with this lack of change in cell morphology. And then when we asked, okay, what does this mean for the calcium levels? And so we found also that the cytosolic calcium levels also change as the cell moves through the cell cycle. And when you don't allow for mitochondrial calcium entry, this doesn't happen either. And the consequence of this is that the mitochondrial respiration doesn't increase during the cell cycle. So these are, again, these are cells at 16 hours after addition of PDGF, and the wild-type cells have started making quite a lot more ATP measured as increased oxygen consumption rate, but the MCU knockout cells just can't keep up. All right, so at this stage, we then asked, okay, so there is something about the cell cycle in MCU in wild-type cells which allows for more calcium entry. How is this possible? So we went back and actually looked at the composition of the complex, which by then, 2 years after we started doing the studies, we learned can be regulated. Okay, so here Olga did Western blots in wild-type cells, which are both arrested and those at 16 hours after addition of PDGF, those cells which are actively proliferating and making ATP, and she found that that the negative regulator of MCU, or one of the regulators of MCU, I should say, MICU is decreased, and so is this other regulator called IMRI, and the negative regulator of calcium influx called MCUB is also downregulated. So with this, we believe we can explain why there's increased calcium influx into the mitochondria at 16 hours, which allows for greater ATP production and progression through the cell cycle. And this is just summarized on this slide, where we found that MCU deletion is sufficient to reduce muscle cell proliferation, and the levels of MCU during the cell cycle actually fluctuates. And this also correlates with mitochondrial shape and metabolic activity. Okay, so then let's move on to the second story, which I believe is the one that you had to be pleasure. I know it was a pleasure to read. So I'm going to give you my perspective on this study, and maybe it's a little different from what you concluded on this. this is the story of Emily, who is an MD PhD, was an MD PhD student in my lab. Is now a PhD student, so moved one step further to fellowship and just had a baby. So she was interested in cell migration, and she also worked on this enzyme. It's the calcium primordial-independent kinase 2, which is a calcium-independent kinase, gets activated by binding calcified primordialin. And we had just heard that ChemK2 actually can phosphorylate MCU and increase calcium uptake. At least there was the hypothesis that this may be the case. So Emily provided some evidence for this by using two very similar approaches. She used cells in which she expressed a peptide inhibitor of this enzyme called Mitochondria, right here, in smooth muscle cells, and she looked for activated, meaning phosphorylated MCU, which is phosphorylated at CA92, to increase the ability of it, which leads to increased influx of calcium, I should say. Okay, so when mitochondria was expressed, she found that MCU is not phosphorylated. On the other hand, when she used MCU knockout cells, she found that chem kinase does not get phosphorylated compared to wild type conditions, but that this can be recovered when she overexpresses wild type MCU, but not when she overexpresses the mutant in which this phosphorylation site is inhibited, in which the phosphorylation site of MCU is inhibited. So the story that we put together with this is that initial calcium influx activates MCU, which activates and phosphorylates ChemK2, which then leads to the activation of MCU, kind of a feed-forward circuit. Okay, so then we measured this in calcium studies. So here we're measuring the calcium response in the mitochondria to addition of PDGF, big response to PDGF in wild type cells, a lot lower when mitochondrial chem kinase is inhibited. And when you overexpress mitochondrial chem kinase, you get more calcium uptake, as you would hope. And if you knock out MCU, there's almost no calcium uptake. And when you overexpress chem kinase on top of the loss of MCU, not much happens, which suggests that for chem kinase to become activated, it needs MCU to be present. Okay, so we made a mouse model, right? So in this mouse model, we're overexpressing this peptide inhibitor, mitochondrial chem kinase inhibitor, mitochemkin, in smooth muscle cells, which is shown here. These mice have less calcium in their mitochondria, and this is the injury. So here, this is kind of a model of balloon angioplasty in which new intima forms in contour mice, but not in mitochemkin mice, right? And so we quantify this. Okay, so Emily looked for cell proliferation. She was a little inhibited, but she was mostly interested in cell integration. And the smooth muscle cells do need to migrate out of this media into the new intima for this new intima to form. Okay, so she ascertained with different models and experiments that if you overexpress mitochemkin, the inhibitor, there is less migration as under wild type conditions. Okay, so now that was interesting, but we just didn't understand how this works. So our next idea came from reading papers in fibroblasts. And in fibroblasts, some smart people have actually shown that when the fibroblasts migrate, the mitochondria do accumulate at the leading edge of the migrating cell. And so we first tested whether this actually happens in smooth muscle cells. So these are smooth muscle cells. Here's a scratch. So here the smooth muscle cells have been removed by a scratch. We add PDGF, and then we image after 2, 6, and 12 hours after the injury. And as you can see, the mitochondria in green do move towards the leading edge, whereas under baseline conditions, they just hang out around the nucleus. Okay, so is this somehow inhibited or changed when you lose MCU? The answer is yes. So here we are overexpressing the inhibitor peptide. We have cells from MCU knockout mice. We give an inhibitor, and every single time, these are control conditions. After 6 hours of PDGF treatment, control conditions, the mitochondria have moved towards the leading edge, but they don't do this if you inhibit MCU, knockout MCU, give an inhibitor of MCU. but not if you do other manipulations which affect cell migration, such as blocking phosphorylation. Okay, so then, somebody said, well, Emily, that's all very nice, but can you actually prove that this is really dependent on the mitochondrial calcium uptake? Well, so the experiment to address this question goes as follows. We have MCU knockout cells. The mitochondria don't move. We overexpress wild-type MCU, and the mitochondria start moving towards the leading edge like nobody's business. And then we overexpress a mutant of MCU in which the calcium uptake is inhibited by mutating the C into R. And here, the mitochondria again don't move. And we did this experiment, we analyzed this experiment, and then we also did wound closure assays. And again, the migration of cells is only recovered when you overexpress wild-type MCU, but not the mutant MCU. Okay, so now we know that MCU regulates cell migration through mobility of mitochondria towards the leading edge, and this is definitely dependent on calcium influx. Okay, so now what do the mitochondria do there at the leading edge? Why would a cell need mitochondria to go to the leading edge? And so the answer to this, or one answer, is that mitochondria go to the leading edge to provide ATP focally for focal adhesions. So the focal adhesions are important for the cell to stick to its basal membrane or to the underlying surface. And as the cell migrates, those focal adhesions will form, and then they will be deassembled, and then the cell moves forward, and then there'll be a new focal adhesion closer to the leading edge, kind of at the front of the cell. Okay, so that's what we're trying to image here. So here, these are images of focal adhesions. They are green at baseline conditions, and then shown in red after 90 minutes of adding PDGF. Under serum-free conditions, the cells should move, and you can tell that the focal adhesions really didn't change very much because the green and the red dots still overlap mostly. Under both conditions, with addition of PDGF, the red and the green dots have dissociated, meaning that the focal adhesions have been modeled. And when you block mitochondrial chem kinase and thereby MCU, the focal adhesions really haven't changed very much. So from this, we concluded that the inhibition of mitochondrial calcium uptake prevents focal adhesion turnover. We did a lot more experiments to show the phosphorylation of particular kinases and focal adhesions and so forth. Okay, so this is all very interesting, but actually, what does make mitochondria move, and why is this calcium dependent? And so these are actually ongoing studies in the lab, and they focus on a funny molecule which sits at the outer mitochondrial membrane. It's called MIO. And MIO can bind calcium. And when calcium binds to MIO, then MIO changes its conformation, and the mitochondria dissociate from the microtubules. And the microtubules actually are necessary for mitochondrial migration. So the mitochondria attached to the microtubules, when they're kind of through kinesin, kind of walk along the microtubules to their final destination. And again, when calcium binds to MIO, then this association will not exist any longer, and the mitochondria are stuck. Okay, and again, this calcium depends. So here we did some experiments where we got some MIO knockout cells. And this is the same mitochondrial translocation experiment. And as you can see, the mitochondria don't translocate at all to the leading edge after addition of PDGF. They're all sitting around the nucleus. And then if you overexpress mitochondrial chem kinase to activate MCU, it has absolutely no effect. And if you block MCU, it doesn't have any effect either. Suggesting that the mitochondrial mobility is dependent on this outer mitochondrial membrane GTPase MIO1, and this is actually calcium dependent. So if you block the calcium uptake into the mitochondria, we believe that there is just more binding of calcium to MIO. That's why if you block MCU, the mitochondria get stuck and can't really migrate any longer. And we did a number more experiments where we actually tested the ability of these cell types to migrate and to actually lead to wound closure. And our conclusion from these data was always that if you don't have MIO at the outer mitochondrial membrane, then there's then manipulating MCU doesn't really have any effect. So from this, we concluded that mitochondrial calcium sequestering supports cell migration by promoting mitochondrial mobility, and that mitochondrial translocation to the leading edge is required for focal adhesion turnover and smooth muscle cell migration, and thereby ultimately to new intima formation. Okay, so in the third small, short piece, I would like to come back to the idea that in diabetes, there's accelerated smooth muscle cell proliferation and therefore increased risk of new intima formation, and maybe also of atherosclerotic plaque formation in the first place. There is pretty good evidence that with all stenosis after balloon angioplasty is far more common. Mitochondrial dysfunction has been described in diabetes for a decade. Many different reports, many different ideas how this might work. There's also some data that this enzyme CAMK2 is activated by hyperglycemia, and some data that MCU actually may be changed if there's high oxidative stress or high diabetes or conditions of diabetes in the cell. So these are, again, experiments that Emily did in the lab where she made mice diabetic. As good as a mouse can become diabetic, right? You put them on high-fat diet and kind of give them some other drugs. And so all I can say is these mice look pretty much like a human diabetic. So they have a resting glucose level of 250 milligrams per deciliter. They're pretty big, they have high cholesterol levels, and they also have insulin dysfunction. And so these mice underwent balloon angioplasty again, and here's the new intima. And I don't know if you remember the previous slide of mice under normal glycemic conditions, but the new intima is a lot bigger than the normal glycemic mice. And if you don't have this, the MCU activator mitochondrial CAM kinase, there is no new intima formation, or at least a lot less. And so then we did cell proliferation assays in many different ways. And we used cells from normoglycemic mice and cells from diabetic mice. And I mean, the stunning finding was that the cells from wild type mice with diabetes treated with PDGF, they proliferated like crazy. And this was inhibited when we used the cells in which this MCU inhibitor is expressed. And Olga, the person who did the experiment, did this with numerous summer students, many times over, with different models, with giving a drug, with knocking out MCU, and it was always the same story. And so which actually led us to ask, so okay, so there's more cell proliferation in diabetic smooth muscle cells, and this is inhibited if you don't have MCU. How is that possible? I should say that there have been decades and decades of studies on why cells from diabetic mice proliferate better, smooth muscle cells proliferate better than their normal glycemic counterparts. And there are many different answers, but there's not one key answer how this works. But our data let us think that it must have something to do with MCU. So in wild type cells, huge proliferation, MCU knockout, no proliferation. So something's up with the MCU complex in diabetes. Okay, so the first experiment we did here is very simple. It's actually to look at the complex composition. Okay, so these are smooth muscle cells from wild type normoglycemic mice and diabetic mice. What do you see? Well, you see that there are all kinds of things that changed. So first of all, the pore forming subunit MCUA is the same, but these regulator subunits were different. And in particular, the negative regulator MCUB, which blocks calcium influx, and the regulator MICU1, which blocks calcium uptake, at least when the calcium levels are low, are a lot lower. So there are some big differences in complex composition. And on the other hand, the activation of those parts which would promote calcium uptake, in particular this MCU-CL92 up here, it's hugely upregulated. And so is the phosphorylation of this regulator. Kim kinase. Okay, so this led us to think that something must be completely wrong with the calcium uptake in these cells. All right, so then Olga embarked on this labor of love by plating smooth muscle cells and measuring the calcium uptake. And so she did this by adding really, really small pulses of calcium, like 100 nanomolar, 500 nanomolar, and so forth. So different concentrations of calcium. And then she measured what this does to the calcium. So in under wild type conditions, if you give very little calcium, not much happens, not much uptake. And then maybe at 2 micromolar, there's some significant increase in calcium in the mitochondria, right? Okay, so now in her diabetic cells, with very small concentrations of calcium, there's already some uptake. And then if you give higher concentrations, there's less uptake. Okay, so this is kind of what you would get if you just plot the different calcium concentrations used in these 5 different conditions. This is the uptake in normoglycemic cells, very low at baseline, a lot better with high concentrations. And in the diabetic cells, well, there's quite some calcium going with tiny concentrations of calcium in the cytosol. And this looks like this, right? And this is kind of another measurement of this. Okay, so what does this look like? Well, this looks like you've lost MICU, right? Because MICU blocks the calcium uptake at low calcium concentrations on the outside, and it allows for more calcium uptake at high concentrations. And that's why in the wild type condition, the curve looks like this. But in diabetic conditions, it looks like that. It looks exactly like what you would expect in a MICU knockout cell. Okay, so we are going through a lot of experiments where we're trying to do 2 things. We're trying to actually mimic, or we are trying to test whether we can mimic the effect of diabetes by knocking down MICU1 in a normal cell. And we're trying to repair diabetic conditions by overexpressing MICU in a diabetic cell. And we're almost there, and this looks very much like this could work. Now, a completely different question that we wanted to ask is actually what happens to cell metabolism. And I should say that a smooth muscle cell at baseline will just have a lot of glycolysis. But they do need, as you've seen in the previous slides, some mitochondrial calcium uptake to maintain particular phases of the cell cycle. Now, diabetic cells grow faster, so do they just are more metabolically active? And how could this relate to calcium? Well, you may remember that calcium influx into the mitochondria drives TCA cycle activity. And it does so by activating some of the key enzymes in the TCA cycle. So we went ahead and gave some cells to our metabolomic core, and this is what you get: a lot of data and a lot of confusion, a lot of headaches. Kind of run to the core and say, gee, what does this mean? Can you tell us? Well, so that's what they told us this means. So what we found is that in diabetic cells in a dish, there are some, so these would be the red ones. There are lots of changes which aren't present, or there are lots of differences between the diabetic cells, I should say, and the normal glycemic cells. And the bottom line of all these changes basically is that there's more TCA cycle activity in diabetic cells. And so one way of looking at this is at glutamate, which is one of the big players in cell proliferation. It's something which gets shuttled into the TCA cycle. And there's a lot less of this around in diabetic cells, which basically means that there's more glutamate getting sucked into the TCA cycle to help with sustaining TCA cycle activity. So when we measure the flux of all these metabolites, which you would do by giving labeled metabolites, and then you measure how much labeled metabolite is still around. And so here also the first impression is that more of this glutamate gets sucked into the TCA cycle and runs around, and then some of these TCA cycle intermediates get shuttled out of the TCA cycle to serve as the building blocks for new cells. Okay. And if you block MCU by giving this inhibitor, this doesn't happen. So here we actually, from this we conclude, and we're still working on this, we have a lot more data to analyze. We conclude from this that diabetic smooth muscle cells have increased TCA cycle activity, which is probably directly related to remodeling of the calcium influx into the smooth muscle cells, which drives the activity of some of these enzymes. And if you just block the calcium uptake, well, then you don't get increased metabolic activity. So here in this part, we found that the deletion or inhibition of MCU inhibits smooth muscle cell proliferation very strongly under diabetic conditions, that there's some remodeling, meaning MCU-B and MICU-1 are reduced in these diabetic smooth muscle cells, and MCU is also phosphorylated, which is more active. Altogether, this means that the calcium entry is increased, and this drives increased smooth muscle cell activity. And all this can be reversed by MCU inhibition. And we have a number of current experiments going on, like where we're trying to, you mentioned, lower MCU and lower MICU expression in normal cells or increase it in diabetic cells to see if we can mimic or cure the conditions of increased smooth muscle cell proliferation in diabetes. So this is the last slide. And so here, so the question of this is all very nice, but what do you do with this, right? This is insight into cell metabolism and cell proliferation, but does this help the patient who we saw on the first slide? Well, so the first good news is that there are some MCU inhibitors which are under development. And in fact, there is already one called RU265, which is cell permeable, and so it's been around for about a year. And we've been using it in a lot of different experiments, but there was just a paper coming out saying that it has neurologic side effects. And when we saw the paper, one person in the lab came and said, oh yeah, I've been injecting these mice and they do these funny movements after I inject them, and they have seizures. So, okay, so this is probably not going to be the right way of treating a human being, which means that we would need to be able to deliver selectively to those smooth muscle cells which are about to proliferate, if we ever wanted to have any chance of making MCU inhibition a viable option as a treatment. So we have a collaborator. I have this next door. I have the guy next door, does these cool things. He makes aptamers. So aptamers are made out of RNA. And so he screens different RNA structures. And some of them that he found bind selectively to smooth muscle cells. He spent 6 years on finding these aptamers. And so these aptamers can be coupled to siRNA, right? So it's an RNA with an RNA, so it sounds like, right, no-brainer. So you couple the siRNA to the aptamer, and then you deliver this and test whether this in vivo will decrease smooth muscle cell proliferation and neo-intima formation. And we're actually about to start testing this, and the funding should set in next month or so. So this is one of the ways how we're trying to actually deliver selectively to those smooth muscle cells that are forming the neo-intima. We will see in a few years or so whether this works or not. I'm going to stop here, out of time anyway. So this is the lab in COVID times, right? So this photo taken a few months ago when we all had to wear our masks. So one of the main players in the lab, you can tell is this person, that's Olga, who did a lot of the experiments, and she works with a number of other people in the lab on this. Benny is a new graduate student, Kim, who's the lab manager in charge of the mice, and there are a number of other people, undergraduate students, right? So people like you last year or a few years ago, right? So we have a couple of undergraduate students in the lab who are trying to introduce to science, and they've made really, really interesting and important contributions to this project. And all the people in red really deserve to be mentioned, and most of them will be on the next paper. I'm going to stop here, and I'll be happy to take any questions.
Yes. Great talk, thank you. I was wondering a little bit about your findings within the knockdown cells, the second originally talked about, and the original mass that you showed from memory, and why there may not be
yes. Yes, so the question was in relation to the mouse that I showed, the white mice of many slides ago, and why there weren't any findings, and mouse models that we're using in the lab. So that's a really good question. So the original mouse, the white ones, they don't have any MCU ever. It's deleted. It's deleted from conception. So I think, and many other people think, that there must be some compensatory mechanisms which the group that published the mouse has been looking into, and well, we haven't, because we didn't think it was the main focus of our work, but we believe that there are some compensatory mechanisms. So the way about this is, if you work in the lab, you might know, so there are some ways to induce recombination and knock out a gene later on by having a mouse in which the gene of interest is surrounded by these LOXP sites. And then you cross this mouse with a Cre mouse, which expresses the enzyme Cre, the DNA recombinase Cre in a particular tissue. And then if you want to be fancy, oh, actually it's not that fancy anymore, you have an inducible model where you give some tamoxifen, and then you get recombination, and then you can study the relatively acute effect of this gene deletion. And that's the model that we're using, also with MCU knockout. Is there a difference? There are big differences. Yes. Yes, 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 yes. Yeah. Yes. Mm-hmm. Yes, so just to clarify, so the diabetes gets induced by people that by putting mice on high-fat diet for 8 weeks, and the mice become pretty chunky, and then we inject a drug which decreases which kills the beta cells in the pancreas at a very, so just give a very low dose. We just lose a few beta cells. And this is kind of what you would get in a human. So the way to induce the diabetes is really a combination of this drug plus high-fat diet. And really we found that in diabetes, the differences in cell proliferation are much bigger than in non-diabetic mice, and which this really led us to ask, why is MCU important in diabetes? So are there other studies on diabetes and MCU? The answer is yes, yes, there are. There have been a few, and they're just coming out. And it's a little, this is kind of related to this other question of, using mouse models to study human disease. So mice don't really get diabetes. And the different models have shown somewhat different, different data, but the studies which are most like ours have shown that there are changes in the heart in MCU function, which would increase the likelihood of a mouse to get arrhythmias. And yes, there's good data that in diabetic patients, there's increased risk of heart failure and also increased risk of arrhythmic events. So there could be a direct connection between MCU and arrhythmias, which really remains to be determined further. Yeah. Oh, okay. So that's a fantastic question. And so I should probably say that, so the question is there any other evidence for MCU in other diseases? So this is a fantastic question. I should also say that about 6 months ago, I talked to one of the people who actually identified MCU. His name is Vamsi Mutha. He's a really good speaker. So he said, well, you know, I've been moving away from MCU because I'm not sure it's really important in human disease. And I said, well, listen, look at our data. We think it is in diabetes. So there are a number of other scenarios in which MCU has been investigated or thought of in diseases. One of them is cancer cells. So cancer cells proliferate, right? And they are pretty uninhibited in their ability to proliferate. And actually there are data showing that the aggressiveness of certain cancers tends to directly correlate with the level of MCU in the cells. Now, does this really say it's X is causing Y or the other way around? No, but at least there's some data that correlates. So there's some data for cancer, there's some data in the heart, there are some data that also the ability of a pancreas to release calcium, to increase, to release insulin depends on mitochondrial calcium uptake. So there may be a couple other disease models in which this may actually pan out. Although initially people were a little bit disappointed. But it's at second look, I think there's a lot more learned if you just really look into how this complex is composed and how it works in different organs.
Thank you so much. It's a pleasure to have you here today. I'm really interested in the proliferative phenotype. So, did you notice any changes in the phenotype of the cells, say when you induce them with PDF or induce them as a potential to look at other therapeutic targets of MCU in general, if we could narrow it down to something specific?
Right. So yes. So I mean, if you have something which is upstream in a pathway, you would, and you're worried that this is going to cause a lot of side effects, then you may think, okay, so then what are the key signaling steps further down in this pathway, which are activated, but I could block them. So yes and no. So we have not really investigated this much further, but you could think that, you know, once we know what is different in the cell metabolism in diabetic cells and, you know, what's differently regulated in MCU knockouts, we may be able to actually pinpoint towards particular reactions regarding mitochondrial metabolism, which may be targeted. Yeah.
Thank you. That's fantastic. You mentioned briefly the therapeutic intervention and something about the behavior of the mice that were jumping around.
Yeah.
And so I'm very curious because the possibilities are vast in my mind in terms of neuronal phenotypes and calcium homeostasis and modulated glucose transfer. Anyway, have you or has anybody looked at kind of the cellular neural phenotype in terms of physiology or various cell biological processes?
So yes, I'm not an expert on this. I have a friend and collaborator at Iowa who is interested in MCU and mostly studies MCU-B. And yes, so for example, if there are particular, so he has induced seizure activity in MCU-B knockout mice, and it is definitely a lot different. So you've lost the negative regulator, therefore you have more calcium influx and the cells do have more, the mice have more seizure activity. So yes, there are data on this. There are a lot of different, you know, subtleties to neurobiology, which escape a cardiovascular biologist, I have to say. But yes, there are data, especially regarding seizure activity. We have also tried to test learning behavior in MCU knockout mice, although we used both white mice, which probably are not the best ones, where we really didn't see much of a difference. But yes, there are a lot of data, or a lot of emerging data in this area. Yes, please.
How did you get the mitochondrial cancers just into the mitochondrial?
Oh, good question. Oh, good. Okay, so many proteins inside the mitochondria are actually encoded by by genomic, meaning nuclear DNA. So the question is, how do these proteins actually get into the mitochondria? And it turns out that they're import sequences. So the import sequences talk to the transporters in the inner and outer mitochondrial membrane, and they actually help direct particular proteins into the mitochondria. So there are mitochondrial targeting sequences, and they are known for those proteins, for or a number, I should say, of nuclear encoded proteins. And you basically, you take this piece of DNA and put this in front of the protein you want to import into the mitochondria. So you can artificially direct proteins into the mitochondria. That's the approach that we use for this. So we use the mitochondrial targeting sequence from a particular COX. And this works pretty well. And actually we did test this, right? So since the protein is transcribed and translated outside of the mitochondria, there's a little bit of this in the cytosol as well. And so we kind of try to estimate how much goes to the mitochondria versus how much stays around in the cytosol. It was probably a ratio of 10 to 1. Yeah, great question.
All right, so at this point, I'll talk to clients. I don't say the paper that students are going to go over is fascinating. I'd like to be there myself and live my mitochondria. So with that, I'd like to thank you.
Thank you.